Welcome to this explainer. Today, we're diving deep into the absolute marvel that is the microscopic manufacturing plant operating inside you right this very second. We're going to look closely at the blueprint of life itself, exploring exactly how cells divide, grow, and perfectly replicate their genetic information. It's genuinely mind-boggling when you think about it. The largest animals on Earth, and even you, started out as just one single cell. So how on earth does that one microscopic entity go on to form the massive, incredibly complex biological structures we see all around us? Well, the secret lies in endless cycles of growth and division, allowing a single parental cell and its offspring to rapidly expand into populations of millions and eventually trillions of cells. Just to keep us on track through some of the heavier terminology, here is our quick roadmap for today. First, the basic cell cycle. Second, exact cell copying. Third, why mitosis matters. Fourth, creating unique gametes. And finally, the power of genetic variation. Okay, section 1, the basic cell cycle. Let's see how a cell gears up for the monumental task of dividing. Get this, if you look at a typical 24-hour human cell cycle, it's wild to think that the actual cell division takes only about an hour. That tiny little sliver of time is the M phase. The remaining 95% of the time is spent in interphase. Now, historically, this was called the resting phase, but honestly, it is anything but restful. The cell is intensely active here, gathering materials, synthesizing proteins, and most importantly, copying its genetic blueprints in a highly coordinated way. And you can clearly see how that continuous cycle of growth works here. We start with the G1 phase, where the cell physically grows and carries out its normal metabolism. Then comes the absolutely critical S phase, or synthesis phase. Here, the actual amount of DNA per cell doubles. So if you start with an amount we call 2C, it jumps to 4C. But, and this is a crucial distinction, the actual number of chromosomes doesn't increase, just the amount of genetic material. Finally, the G2 phase acts as a final prep step, synthesizing the specific proteins needed right before the cell commits to the dramatic M phase. Before we look at the division process itself, we need to clearly define the split, because it's really a 1-2 punch. Karyokinesis handles the incredibly complex division of the nucleus, while cytokinesis handles the physical division of the cell cytoplasm. Basically, first, you neatly divide the identical genetic material, and only then do you actually pinch the outer cell completely in two. Moving right along to section 2, mitosis, which is all about exact cell copying. We often call this equational division, simply because the number of chromosomes in the parent and the new progeny cells remains exactly the same. During prophase and metaphase, it's like a factory assembly line. The machinery untangles its chromosomal material and perfectly aligns those duplicated chromosomes along the middle of the cell. In prophase, that tangled mess of replicated DNA from interphase condenses into neat, tightly packed structures. By the time we hit metaphase, the nuclear envelope has completely dissolved. Tiny spindle fibers attach to these disc-shaped structures on the chromosomes called kinetochores, and they literally pull them until they're lined up perfectly along the equator, which we call the metaphase plate. It is a stunning display of cellular logistics. Then, in anaphase and telophase, those sister chromatids are ripped apart, ensuring each new daughter nucleus gets an exact, identical copy of the genetic blueprint. In anaphase, the centromeres suddenly split, and the individual chromatids are dragged to opposite poles of the cell. Finally, in telophase, the chromosomes reach their destinations and start to decondense back into a loose tangle, while a brand new nuclear envelope builds around each set. Right after this, cytokinesis steps in, either creating a furrow in animal cells or building a new cell wall in plant cells, leaving us with two pristine, completely independent cells. Which brings us to section 3. Why does mitosis actually matter? You might be wondering, what's the grand purpose of this relentless duplication happening inside you right now? Well, mitosis is the ultimate repair and growth mechanism. It's constantly working to replace the cells of your upper skin or heal a scraped knee, ensuring multicellular organisms actually survive. See, as a cell grows, it eventually disturbs the delicate balance between its nucleus and its cytoplasm. Mitosis triggers a division to restore that vital ratio. Beyond that, it's just pure maintenance. The lining of your gut and your blood cells are constantly being replaced by mitotic division. And in plants, specialized meristematic tissues use mitosis to ensure continuous, lifelong growth. All right, section 4, meiosis and the creation of unique gametes. Now, if mitosis is about perfectly maintaining the status quo, meiosis is about shaking things up for the next generation. Unlike mitosis, meiosis is a reduction division. It's specifically designed for sexual reproduction, and it cuts the chromosome count squarely in half to create gametes. Think about it. If two specialized gamete cells, like a sperm and an egg, are going to fuse together, they each need to carry only half a set of genetic instructions. Otherwise, the resulting offspring would have double the normal amount of DNA, and that just wouldn't work. Meiosis cleverly solves this by going through one round of DNA replication, but two distinct rounds of cell division. And the true magic happens right here, during prophase 1. A process called crossing over acts like a microscopic R&D lab, remixing the genetic deck so that offspring are totally unique. During this incredibly complex first phase, homologous chromosomes pair up in a process called synapsis. They form physical links, and through the action of an enzyme called recombinase, they actually swap sections of their genetic code. It's this elegant, totally natural exchange of material that basically guarantees no two gametes are ever exactly alike. Following that crazy genetic swap, meiosis I separates the paired homologous chromosomes into two new cells, which dramatically reduces the chromosome number. Notice how the whole recombined chromosomes are being pulled apart in anaphase I, unlike in mitosis where the sister chromatids split. At the end of telophase I, we are left with two cells that now hold half the original chromosome count, even though the DNA itself is still in a duplicated state. Then we jump right into meiosis II, which functions much more like a standard mitosis. It splits the sister chromatids and ultimately results in 4 entirely unique haploid daughter cells. After a brief resting period called interkinesis, where there's no new DNA replication, these two cells from meiosis I divide again. The centromeres finally split during anaphase II, and by the end of telophase II, the overarching goal of meiosis is completely achieved. You have 4 genetically distinct cells, each primed with half the standard genetic material, ready and waiting for the fertilization process. Let's wrap up with Section 5, the sheer power of genetic variation. The cellular mechanics we just looked at might be tiny, but their implications are species-wide. The crucial takeaway here is that while mitosis keeps you alive day to day through perfect cloning, meiosis introduces the essential genetic variations that keep an entire species evolving over time. It is a brilliant biological paradox, honestly. By purposefully reducing the chromosome count by half, meiosis is exactly what conserves the total chromosome number when those gametes eventually fuse. At the exact same time, because of that amazing crossing over process, it continuously injects fresh genetic variability into the population. Without this microscopic reshuffling, the whole process of evolution would literally have no raw material to work with. It is a nearly flawless microfactory most of the time, but it leaves us with a really fascinating question. What happens to the organism when this relentless, invisible machinery accidentally makes a typo? While these processes are incredibly coordinated, the sheer volume of divisions happening inside you right now pretty much guarantees the occasional error. Sometimes a cell catches the typo and halts the whole cycle, but other times, that typo escapes the checkpoint. It can spark the beginnings of uncontrolled growth or, on the flip side, introduce a completely unexpected new evolutionary trait. Understanding the strict rules of this cellular microfactory is really just the very first step toward understanding what happens when those rules are broken.